Okay, welcome to this evening's Perth ISCAST meeting. So it's my pleasure to introduce our speaker tonight. Dr. Jonathan Gauntlett has modestly given me a very brief bio. Jonathan has a PhD in something genetic and biological. You can probably give us more detail when you start speaking. From the University of Otago in New Zealand. And he's been modifying genes in one organism or another for about 20 years. Jonathan tells us, you can tell us a little more as the talk goes on. Over to you, Jonathan.
Thank you, Peter, for your introduction. Sometimes it's amazing to think that I've managed to make a living for the last 20 years. But thank you also for coming out this evening. I hope that you enjoy listening to it as much as I've enjoyed putting it together, and I'll let you ponder exactly what that means. So we're here to talk about gene editing. Where are we headed? Both the science and also some of the ethical considerations. And so I want to cover this evening, some definitions. What is gene editing? And then look at some contemporary or perhaps the most advanced examples. Not what's up and coming, but what's already here. I think there's a little bit of talk about various things that are maybe not so realistic just yet. They could potentially come in the future. So we'll just look at what's happening now. And then look at different types of, so give an example of the different applications, I suppose. So somatic editing, germline editing, xenotransplantation, and then throughout the talk there's various reflections and a little bit of theological consideration as well. And any opinions that I express during the course of this talk are purely personal in nature. So what is gene editing? Well, gene editing is a method for making specific changes to the DNA of a cell or organism. It can be used to add, remove, or alter DNA in the genome. And so there's a number of different techniques for modifying the sequence of DNA, and I want to focus on what's called CRISPR-Cas9 technology tonight. And then there's gene therapy. So gene therapy is a medical approach that uses genetic material to prevent and treat disease. The technique allows healthcare providers to treat certain conditions by changing your genetic makeup instead of using traditional treatment methods like medication and surgery. So we're going to look at the intersection of CRISPR-Cas9 and gene therapy, because there's other ways of doing gene therapy other than just genome editing. And I'm also going to focus on human examples and not talk about gene editing in other animals or other organisms. So what is gene editing? Well, molecular biologists, for a lot of people it's like a black box, what on earth do they do? But molecular biologists just utilize and adapt tools which are found in nature. So the tool that we're talking about for genome editing, the Cas9 enzyme, is a protein that comes from the bacterium Streptococcus pyogenes. And so bacteria have this protein naturally existing there as a mechanism by which they can defend themselves from viruses. So bacteria like you and I can also be infected by viruses called bacteriophages. And they don't have a complex immune system like we do, but what they do have in its place, although it functions completely differently, is this CRISPR-Cas9 system. And so what happens is that they have in their genome a series of repeats which are strung together, and those repeats derive from the sequences of viruses. And those repeats are expressed and the RNA is expressed. And there's also this protein which combines with the RNA. And that enables the protein to recognize the genetic sequence of incoming viruses. And what it does is when it recognizes the sequence of incoming viruses, it then cuts that viral DNA to pieces, or viral RNA, so that the bacteria is safe from infection. And so what you have is this Cas9 protein, and it can be used by molecular biologists as a programmable endonuclease. So what that means is that you can program it to cut DNA at any specific location in any genome that you like. Not quite, but almost. And that's simply done by hijacking this tool which the bacteria originally used to protect itself against viruses. So what you do is you provide the protein with a piece of RNA, which is complementary to the sequence of the DNA that you want to cut, and the Cas9 protein locates that sequence in the genome and matches it up with the RNA template that it has, and that's where the endonuclease activity of the protein comes into play, and the double-stranded DNA is cut at that location. So in theory, what this allows people to do is, if you can introduce this Cas9 protein into a cell, it means that you can create a break or a cut in the DNA at any location within the genome. And so what happens to the cell when it has a double-strand break is it now is in a crisis. It needs to be able to repair that DNA damage if it's going to replicate its DNA and divide and survive. So there are naturally existing mechanisms to repair DNA damage, because we are encountering DNA damage all the time from UV radiation or oxidative stress or other things that go on with DNA. And so you've got this double strand break at this specific position in the genome. And so now what the cell is trying to do is it wants to repair this DNA damage. And there's a number of different pathways which exist within cells to repair the damage. So there's non-homologous end joining, and that's a process which results in mutations being introduced into the DNA. Usually small mutations, either small deletions or small insertions. There's microhomology-mediated mismatch repair, microhomology-mediated end joining, and it also makes small mutations. And then there's another pathway called homology-directed repair. And homology-directed repair looks for sequence which is the same as the sequence which has been damaged and matches those sequences up, and then uses that matched up sequence to repair the damage. And if you do it via that pathway, you don't introduce any mutations into the genome. Or alternatively, if a sneaky molecular biologist comes along and provides another piece of DNA which has the ends the same as the sequence which has been cut, it can anneal with the ends and then introduce the new DNA into the sequence which has been cut. So as a result of the double strand break, you can end up with all sorts of outcomes. Sometimes the DNA is repaired so that there's no mutations. Sometimes it's repaired in a way which results in small mutations. Sometimes it can result in very large mutations, and sometimes it can be used as a way of introducing new sequence into the genome. And so by using this technology, molecular biologists are able to disrupt genes if the mutation is introduced, which results in the gene not being able to function normally. Or you can introduce new sequences into genes, and those new sequences could come from anywhere, because you're able to synthesize or clone any DNA sequence essentially that you would like. So in the term of genetic engineering, this is commonly referred to either a knockout or a knock-in. If you knock out a gene, you disrupt the function of the gene. If you knock in something into a gene, you're introducing DNA from somewhere else into the genome at that position. And I think it's very important to make a distinction at this point in the talk, because it's an important distinction for genome engineering altogether, and as to what cell you're making the mutation in. So, often people will make mutations in cell lines, and that's all it ever intends to be, it's just a cell line. But if you were to make a mutation in a cell and then introduce the cell into the body, or if you were somehow to introduce the CRISPR-Cas9 endonuclease into a certain location in the body, either by viral infection or by breathing in lipid particles or something which contain the CRISPR-Cas9 components, you might introduce genetic modifications into particular cells within the body. And so that will change the genetic makeup of those cells. But the key here is that it's not changing the genetic makeup of the cells which are responsible for passing on your genome to the next generation. And the cells which are responsible for passing the genome on to the next generation are the cells in your testes or the cells in your ovaries. And if those cells that produce eggs or produce sperm are genetically modified, then that genetic modification can be passed on to the next generation, and then that individual is completely genetically modified, and they will then pass that on to the next generation, You've introduced that genetic modification into the gene pool. at that point. So it's quite different in terms of the consequences, how you introduce, where you introduce the genetic modification. If it's just what's called somatic gene editing, then it's just like your liver or your lung or your skin cells or your neurons or whatever. It doesn't get passed on to the next generation. It just affects that individual. But if it's done in an embryo, then all the cells in the resulting organism are modified, and consequently, so will the germline, and the genetic modification will be passed on to the next generation. And then you've introduced that genetic modification not just into one individual, but the offspring of that individual as well. A modern example of somatic gene editing is the recently FDA-approved, the first ever FDA-approved gene therapy, which is using genome editing and CRISPR-Cas9 technology. So it's a technology which is treating sickle cell disease. And so sickle cell disease is caused by a mutation in the hemoglobin beta subunit gene, HBB. And in the red box there is the actual DNA base, which is mutated. That's part of a codon which encodes glutamic acid at position 7, and it's mutated to a valine at position 7 of the human beta subunit, hemoglobin beta subunit. So if an individual carries this mutation, and so everybody has got 2 copies of a particular gene, so just say there's 2 alleles at any given loci. If the person has 1 mutated copy and 1 normal copy, that person is healthy, but they're also resistant to malaria. Well, not quite resistant, but they have reduced sensitivity or susceptibility to malaria, and the consequence of getting malaria infection is a lot less. But if you have 2 copies of the mutated gene, you have sickle cell disease. And so hemoglobin beta is an important protein. It forms one of the subunits of hemoglobin along with hemoglobin alpha. And it's responsible for transporting oxygen around the body within red blood cells, and it's the main component of red blood cells. So when you have this mutation in the hemoglobin beta subunit, it changes the structure of the hemoglobin protein so that red blood cells become deformed, and they adopt a sickle cell shape. And they're not so good at carrying oxygen around the body, and also they're not so good at traveling through blood vessels, so they can get stuck. And so people with sickle cell anemia suffer a number of consequences as a result of this. They have pain crises where various organs are not receiving enough oxygen, red blood cells are getting stuck in the veins, and they also have anemia, which is just saying they don't get enough oxygen. There's swelling, and they're more susceptible to infections, vision problems, stroke. I haven't gone to the right slide, have I? It's changed the formatting a little bit. And other complications as a consequence of being starved from oxygen temporarily. So researchers have looked to see if they can use CRISPR-Cas9 genome editing to treat this disease. And so, as I said before, it's the world's first FDA-approved genome editing therapy using CRISPR-Cas9 technology. And so what they've actually done is they haven't targeted the human beta-globin gene directly. The thing is that hemoglobin is a little bit interesting in that when you're an adult, your hemoglobin is made from the alpha subunit and the beta subunit. But when you're an infant, it's made from the alpha subunit and the gamma subunit, and the beta subunit's not expressed. And the reason why fetal hemoglobin is made using the alpha subunit and the gamma subunit is because fetal hemoglobin needs to have higher affinity for oxygen than adult hemoglobin, because you have to steal the oxygen from your mum through the placenta, from the mother's bloodstream. And so there's the specialised hemoglobin gamma subunit. And when shortly after you're born, or a few weeks after you're born, and the chart showing that there, that the beta hemoglobin levels start to come up, and the gamma hemoglobin levels start to come down. And the reason for this change in the regulation of the genes is because of a DNA binding protein, which binds within the regulatory regions of these genes, called BCL11A. And that gene represses the expression of fetal hemoglobin, the gamma subunit. And so what the medical people are able to do is they harvest hematopoietic stem cells from a person who has sickle cell anemia. And they introduced the CRISPR-Cas9 genome editing tools into those stem cells, and that makes the cut into the DNA at the position of this gene, which regulates expression of the fetal hemoglobin, the BCL11A gene. And that cut disrupts the activity of that gene and prevents that gene from being expressed. So this regulator, therefore, is not expressed. So then that means that the levels of fetal hemoglobin don't come down, they stay up. So you've got this hematopoietic stem cell, which is now genetically modified. And hematopoietic stem cells are stem cells which, when inserted back into the body, make their way into the bone marrow, and they are like the originator cells for all subsequent blood cells, red blood cells, white blood cells, whatever. So they begin the blood cell lineage, the hematopoietic stem cells. So they take the hematopoietic stem cells out of the patient, they genetically modify them, they check that the genetic modification is made. At the same time, the patient is then given a radiation to kill off their normal hematopoietic stem cells, which are left behind. And then the genetically modified stem cells are cultured in the lab, and when there's sufficient numbers of them and the patient is ready because their own red blood cell hematopoietic stem cells have been depleted, they are reintroduced into the patient. And they colonise the bone marrow, and hey presto, now all the blood which is being made from the bone marrow in that person originates from those genetically modified stem cells. And that means that those stem cells, as a consequence of the fetal hemoglobin, the gamma subunit gene being dysregulated, they now are able to express the fetal hemoglobin. So they will have still the defective beta subunit, but as well as having the defective beta subunit, they also have a functional gamma subunit. And that means that they behave more like the people who carry the disease. So they're physiologically healthy, and they're probably still somewhat resistant to malaria. So it's an interesting story because it's thought that the point mutation in the hemoglobin beta subunit has been selected for over the period of evolution in regions like sub-Saharan Africa where malaria is prevalent, and being a carrier of the mutated gene gives you a selective advantage in that you're much more able to survive malarial infection. And it's just an unfortunate consequence that when it comes together in two copies in one individual, that the disease state arises. And the reason why people are more resistant to malaria when they have sickle cell disease is because the malaria parasite, which replicates within inside red blood cells, is not able to do so as well in the misshapen, deformed cells. So the progression of the disease is significantly slower in a carrier than in a normal person. So the thing about this is that it was a long, complicated process in development. CRISPR-Cas9 has been around for 10 years, and people are saying, well, this is the first one now after 10 years, and that's really quick. I'd hate to think how many millions of dollars of investment or funding has been required to do this research. And also, it's a long, complicated cure. I mean, you have to undergo radiation, you have to go hematopoietic stem cell transfer. It's not a straightforward process for the person undergoing it. And so as a consequence, it's quite expensive. So there's a lot of clinical trials currently underway for CRISPR-Cas9 genome editing to treat other diseases. These include cardiovascular disease, blood cancer, solid cancers, haemophilia, inherited eye diseases, metabolic disorders such as diabetes, which I think is an interesting one, having a brother with type 1 diabetes, muscular dystrophy and HIV, and there's a whole list of them at the website given there. But it raises, to my mind and to many other minds, a few questions, because as I mentioned before, it is very expensive. And to have this treatment, if you happen to have sickle cell disease, it's going to cost you at least $2 million US. So it's not like your government is going to start funding that. So you're going to have to start crowdfunding pretty much now. So the thing is that the cynic in me says, well, you know, it's just big business making more money. It just increases the difference between rich and poor. But then the pragmatist in me says, well, these companies have had to spend millions and millions of dollars investing money to develop this technology. It's a complicated technology, and so they need to have a financial incentive to put that investment forward in the first place. But it's still an interesting point, I think, about the potential for inequality, because it's nothing new, inequality in the outcome of healthcare. I have a chart there on the screen which graphs the amount of money spent on healthcare along the x-axis, and I believe it's a logarithmic scale. I can't quite see it. And on the y-axis is average life expectancy for that country. So it's kind of using average life expectancy as a proxy readout for quality of healthcare in those given countries. And you can see that there's quite a good correlation between the amount of money that a country is spending on healthcare and the average life expectancy for people in that country. And at the top right-hand side there, you can see countries like Australia spends a lot of money in healthcare and gets the result of higher living expectancy, perhaps as a consequence. It's interesting there that the United States spends about 5 times more on healthcare than Australia, and yet they have a lower life expectancy. And I'm just speculating, but I hypothesize that it might be because the private healthcare system in the United States is very, very good for those people who can afford it, and very, very bad for those people who can't afford it. So you get a lot of disparity in outcomes. And then you see coming down through the middle, countries like China, I've just picked a few, Indonesia, Afghanistan, Bangladesh, and then down the bottom there you've got some of the African countries which don't spend so much money. And you know, South Africa, poor South Africa spends a lot of money there, it doesn't have such good outcomes. And so I wonder whether maybe the HIV pandemic has a role to play in that. But anyway, so Francois Baylis has said unequal access to genome editing technologies will both accentuate the vagaries of the natural lottery and introduce an unjust genetic divide that mirrors the current unjust economic and social divide between rich and poor individuals. So he's envisaging a future where genome editing technology is available for those who can afford it. You know, if you happen to be a billionaire's son or something or other and you're born with sickle cell disease or any other of these diseases, that's fine. You can go ahead and get treated and you can live a healthy, normal lifestyle. Whereas if you're born the son or daughter of a low-income family, well, good luck to you. And so, you know, that already exists with sickle cell disease. There are ways of not, you know, not curing it, but there are ways of managing the disease. And this would be aided if there was some more funding for some of these sub-Saharan countries where there's a lot of sickle cell disease, but there isn't the money there. And so the treatment that was given to the first patient was, of course, to an African American. Anyway. So that's the contemporary example of somatic gene editing, where in this case, it's just the cells which give rise to blood in the body which have been genetically modified to heal that person of the disease, and they won't pass that genetic modification on to their descendants. But there has been at least one example that we are aware of germline genome editing. And this occurred back in 2018, and it caused a bit of a stir at the time when He Jiankui in China became the world's, the first person to introduce a genetic modification into the human gene line by genome editing 3 baby girls, or I should say 3 embryos, which later became 3 baby girls. So the father, who donated sperm, was an HIV positive person. Although the sperm was washed, and I don't know what that means, but apparently the technique means that there's little to no risk of HIV being passed on to the baby through the sperm. And then the mother did not have HIV. So embryos resulted from in vitro fertilization, and then I think they were electroporated, so with the CRISPR-Cas9 technology. So electroporation means you take the embryo, you subject it to an electrical field, which transiently opens up pores within the membrane of the cells in the embryo, and that allows for the CRISPR-Cas9 components to enter into the cell, and once the CRISPR-Cas9 components have entered into the cell, they can cut the DNA at the desired location that's been programmed by He Jiankui. So what he was targeting was the CCR5 gene. And the thing with CRISPR-Cas9 is, as I said before, it makes a double-strand break, but the double-strand break can be repaired via any number of ways. So I don't know how many cells were in the embryo at that point in time when he introduced the CRISPR components, but let's say there were 8 cells in that embryo. They may not have all gotten the CRISPR-Cas9, so some of them might be still wild type. Some of them might have got the CRISPR-Cas9 and made a cut and a deletion occurred in the gene, or maybe an insertion occurred in the gene. Or maybe the cut didn't occur in the CCR5 gene, but maybe it occurred somewhere else in the genome where it wasn't intended to cut, and there was a mutation at that location in the genome. So it could be that the children which develop from these embryos are not all the same genome throughout the whole body, but some of their cells which are derived from the one that didn't get mutated are normal. Some of the cells which derive from the cell which resulted in the deletion of the gene have a deletion for that gene, or an insertion in that gene. So that's called a mosaic. That person would be the mosaic for that genetic modification. They could have any number of different alleles in any different particular cell lineage. So Hei Jiankui was widely condemned for this action. So there were lots of questions raised about it. There's a questionable activity to do this. Did the participants in the study give informed consent? Did the mother and father really know what they were doing? And did the children give their informed consent? That's one of the things about germline editing, is that, of course, children can't give their consent to inherit a genetically modified genome, and neither can their grandchildren and their great-grandchildren, and so forth and so forth. So that's one of the ethical questions about introducing genetic modification into the germline. What will be the long-term effects of making this genetic modification? We think we understand a certain amount about what the CCR5 gene does, but do we understand it fully? Most likely we don't. And so the argument is that it's irresponsible to use premature technology. We know that CRISPR-Cas9 will make the cut in the genome at 99.9% of the time at the right location, but 0.1% of the time it doesn't. What happens then? What happens if you're a mosaic? What happens if the gene has other functions that we're not currently aware of? What is the potential outcome to the person from receiving that genetic modification? He did no preliminary studies. He didn't do this experiment in animals first. He didn't do this experiment in cell culture first. And so he just was like, all right, here we go. We'll straight off the bat into a human. That's the sensible starting point for this experiment. And so I've already mentioned the off-target effects where it cuts in another location in the genome. So it turns out that in doing this, he wasn't complying with the national institutional ethical guidelines at the institute he was working in China. And I think maybe because of the consequence of breaking the rules, he served 3 years in prison. And people were also asking, what's the point? If you're going to genetically modify, the human germline, maybe you do so in a disease which is, I don't know, like a horrible, debilitating childhood disease. I went to a conference the other day where the guy was looking at using CRISPR-Cas9 genome engineering to treat muscular dystrophy or something, I can't remember, and it occurs in babies. And that person is affected for the rest of their life and has a shortened life expectancy. Maybe you choose a gene like that. But no, we'll talk about what he did choose. He chose to confer resistance to a disease which is entirely preventable. Why would you choose to confer resistance to a preventable disease when you could just prevent that disease without having your genome modified in order to do it? And he wasn't trying to protect the child from the HIV from the father because the sperm was washed, so there was little risk of that. So the preventable disease is, of course, HIV. So he targeted the CCR5 gene in the human genome, and CCR5 is a chemokine receptor, and chemokines are signaling molecules which are involved in the human immune system. And I think this receptor picks up inflammatory cytokines. So it plays a key role in driving the immune response. And the immune response is so insanely complicated. Don't ask me any questions about it. I cannot give you any answers. So it's expressed in T cells, which are an important cell in the immune system. And HIV infects the T cells in the immune system. And this is why HIV is so good at escaping the immune system, being able to persist latent within a person for a very, very long time. And so in order to get entry into T cells, the virus has to bind and interact with cell surface receptors. And the main cell surface receptor it binds to is CD4, and a lot of T cells, especially active T cells, have got this CD4 receptor. But in order for this interaction between the virus and the CD4 receptor to be stabilised, this is where CCR5 comes into play. It stabilises the interaction between the CD4 receptor and the HIV virus. So usually CD4 would be picking up an antigen which is being presented by an antigen-presenting cell, such as a macrophage within the immune system. But HIV has a protein on its cell surface, GP120, which can mimic what that looks like. And so CD4 latches onto that as well and gets tricked into introducing HIV into the cell. So it is known that people who have a mutation in CCR5 are resistant to HIV. And in European populations, as many as 1% of people carry the natural mutation in the CCR5 gene. And the only people who have ever been cured of AIDS, AIDS is treatable and manageable, but you can't cure it as yet. The only people who have been actually cured of AIDS are people who have, again, the hematopoietic stem cells, received hematopoietic stem cells from people who naturally have this mutation. And then they express T cells which have got the mutated CCR5 gene, and HIV can't gain entry into the cells. And there's a caveat to that because HIV is a bit sneaky. It uses CD4 and CCR5 in early stages of infection of the disease, but in later stages it switches receptor. So it's not necessarily going to be 100% protective even if you do have this mutation. So, you know, in He Jiankui's defense, he wasn't introducing a completely novel genetic mutation into the human genome. It's a naturally occurring mutation. It's just that there's no guarantee that CRISPR-Cas9, when the DNA is repaired after the double strand break, would have made exactly the same mutation. It could have been any sort of type of mutation, but you could perhaps argue that given that it's a naturally occurring mutation, we sort of know what the consequences of that mutation will be. But the thing is we don't. Because genomes are complex interacting systems. One gene doesn't stand in isolation. So one gene could actually produce multiple different proteins which have different functions. One gene could be expressed one way in one organ and a different way in a different organ and have two different functions in different organs. And gene products interact with each other. CCR5 interacts with CD4. But that's probably not the only thing it interacts with. And so CCR5 is not only expressed in T cells with the immune system, it's also expressed in microglia in the brain. So what is the effect of knocking out CCR5 in the whole body? If it's not only knocked out in T cells, it's also knocked out in your brain. And so it's also expressed in microglia, which are an important immune cell within the brain. And so CCR5 mutations, because it's important in the immune system, it changes resistance to a number of other diseases, so increased resistance to smallpox, COVID, dengue fever, and Zika virus, but increased susceptibility to influenza and West Nile virus. And as I was saying before, it's also expressed in the brain, so subsequent to He Jiankui's mutation, there was a study which showed that stroke victims show improved neural plasticity. I guess that's like the ability to recover or rebuild neural networks after a stroke. So could the girl's brain development have been affected? Well, if so, maybe no differently to other people who carry the mutation, but it's more than just HIV infection, you see, that's the point. And it highlights how we don't fully understand the complexity of the genome. When we make a change at one point in the genome, what is the knock-on effect in other areas within the organism? So I mentioned before in the sickle cell disease that they knock out the BCL11A gene, which prevents the fetal hemoglobin from being repressed, so that fetal hemoglobin is expressed. And that's fine because you've only knocked out that gene in hematopoietic stem cells. But if you were to knock out that gene in a whole body, so that it wasn't expressed in the brain, well, BCL11A is a DNA binding protein. It doesn't just regulate the expression of fetal hemoglobin. It regulates the expression of a number of other genes which are expressed in the brain as well. And so it's been shown to be an important regulator for axon outgrowth and branching. So if you were to make that same knockout which cures a sickle cell disease patient in an embryo and have it expressed in the whole body, that person could have their brain development impaired. They could end up sort of mentally disabled in some fashion. So it just, it raises the question of, do we know what we're doing, right? So people have said, germline editing is such a wonderful opportunity. And this is where it's potentially a bit science fiction as well, because there's this possibility of removing all these cruel diseases from the human germline. I'm on the spot now. I have to think of some genetically inherited diseases, like Huntington's disease and whatever else. There's any number of nasty genetically inherited diseases. And if we could germline edit all of these out, we could remove these diseases from the human gene pool, and wouldn't that be utopia? So, it's potentially a fantastic opportunity to reduce human suffering. But questions have to be asked whether it's eugenics. Are we trying to artificially enhance the human gene pool? And then people raise issues like, designer babies. Can I give some embryos to a scientist who then produces a cocktail of genetic edits within those embryos, so that my child is intelligent and witty and funny and has blue hair, blue eyes and green hair? The thing is that while that's an interesting question, it starts to get to the point of science fiction rather than science fact, or potentially science way in the future rather than science now. And I might explain that a little bit later on. So while some of these questions are a little bit, some of these potential possibilities and the questions they raise are at this point in time where the technology is still nascent, perhaps a little bit sensationalist or a little bit premature. So then, I guess the yin-yang to the opportunity of removing human suffering and treating human disease is other questions like, is it hubris? Are we playing God by editing the human genome? Do we understand what we're doing? Should we do it even if we did understand what we were doing? Is the human genome so important that it's sacrosanct? Does it make us who we are to the extent that we shouldn't play around with it? And so Article 1 of the Universal Declaration of the Human Genome and Human Rights, as declared by UNESCO, and it's interesting that these people have been thinking about this, there's plenty of funding for bureaucrats, it seems, which is not to say that there isn't a role for this. The human genome underlies the fundamental unity of all members of the human family, as well as the recognition of their inherent dignity and diversity. In a symbolic sense, it is the heritage of humanity. Well, I don't know if that makes sense either. Is the human genome static? All genomes are evolving. There was never a point in time when the human genome hasn't been evolving and has been static. It's not static today, and it never has been, and it never will be. What even is the human genome? I'm human, Jake's human, you're human, but I don't have the same genome as you. My genome, if I was to match it up to you, I would expect to see millions of base pairs of difference. My children, they don't have the same genome as me, 50% me, but they have at least 60 mutations, which are either inherited from me or from my wife. They're unique mutations which arose somewhere in the development of my children. So they're different too. And then what is the human genome? We have a reference human genome sequence. For any given gene, this is the reference sequence you can go to. This is ATGC, GC, ATGC, right? But where did that even come from? Well, 2/3 of the reference human genome came from one single white male who went into a blood donation clinic in Buffalo, New York at the beginning of the Human Genome Project. So it hardly represents all the diversity, genetic diversity of all humans, from the deep jungles to the mountain peaks, to the deserts, or wherever we happen to live, and all the different shapes and colours and sizes that we come in, is not represented by the reference human genome. So what even is the human genome? And when making a genetic modification, who decides what's desirable? Who decides what's normal? And what about genetic diversity? I mean, shouldn't we all be a little bit different to some extent? Isn't that part of what being human is? Isn't that a good thing, so that when COVID comes around, it doesn't kill us all? Because some of us are naturally resistant because we have a CCR5 mutation or something like that. So, interesting questions about it. So, moving on from somatic genome engineering, where you modify a cell that doesn't get passed on to the next generation, to germline editing, where the genetic modification is passed on to the generation, to xenotransplantation, where you introduce the genetic modification in another organism and then you bring that into your body. So it's about organ transplantation and organ donors. So there's many disorders that can only be treated by organ transplant. And, end-stage renal disease, you need a new kidney, you need a new pancreas, you need a new liver, you need a new heart, you need a new lung. You can survive without a big toe, but there's a lot of things that, if your lung starts to give way, you might be had it unless you can get a donor that gives you a new lung. But there's a shortage of lungs and hearts and livers and pancreases out there, and 20% of people who are on a waiting list will die while waiting for an organ. So it's a bit of an issue, and so there's research going on as to, well, where can we get organs for these people who need organs so they can continue to live. And pigs would be an ideal organ donor because pigs are the same size roughly as people with similar physiology. So a pig heart will slot into your chest and attach fairly nicely, apparently. But there's significant barriers because your immune system will recognise that this pig heart, which has been introduced into your body, is not your heart, it's a pig's heart. And so there's a hyperacute immune response to begin with because pig hearts have carbohydrate moieties on the surface, which are instantly recognised by the human complement system, which is a segment of the human immune system. And within hours, that pig heart will be being attacked by the immune system and destroyed. So there ain't no point putting a pig heart in you if you're only going to survive for an extra couple of hours. You probably haven't even woken from surgery. What we can do then is we can genetically engineer the pig so that it's a better donor for a heart or a liver or a pancreas or a spleen, but not big toes, no. Don't need big toes, apparently. So what you do then, and just from reading the literature, I haven't got any personal experience when I say what you do. You can make genetic modifications into the pig genome in the genes that express these carbohydrate sugars on the surface of the cells of the heart. So there's nothing there for the immune system to recognise. So you go in with your CRISPR-Cas9 genome editing and you knock out those genes so then now the pig is not going to express those. And you can also not just knock out the pig genes, but what if you introduce a couple of human genes into the pig? And those human genes will help, they're genes which regulate the human immune system, that regulate the complement system. And so you can express genes which will tone down the human immune response to those tissues that are expressing that gene. So you can use CRISPR-Cas9 to remove pig genes, and you can use CRISPR-Cas9 genome editing to introduce human genes. And so what you do is you take a pig skin cell and you introduce the CRISPR-Cas9 components into it by electroporation, and the CRISPR-Cas9 system goes and it deletes the genes that you want deleted and introduces genes that you want introduced. And that's not straightforward because the more modifications you have to make, the more you need a single cell to have all of those modifications, not just 2 or 3 of them. If you're making 6 modifications, it has to have all 6. And CRISPR-Cas9, because it cuts DNA, is good at destroying genes, but it's not so good at introducing new genes. It can, but it's less efficient. So then I would imagine you'd have to screen a lot of cells before you identify one which has got all the edits that you want in the right place. But once you've identified one, you go, right, this is the genetically modified cell. I'm going to take the nucleus of that cell, which contains all the DNA, and I'm going to take a pig zygote or a pig egg, one or the other, I'm not sure exactly which. I'm going to remove the nucleus from that egg, pig egg, and I'm going to replace it with the nucleus of the skin cell that I've genetically modified. And so then this egg cell has now got the DNA in it that's been genetically modified, and that's called somatic cell nuclear transfer. And then you can stimulate that egg cell to start dividing, and 2 cells, 4 cells, 8 cells, 16 cells, and hey presto, you've got an embryo on the way, so we'll introduce that into a surrogate pig mother. And that embryo will develop inside the mother, and a pig will be born, and that piglet will be genetically modified. All of the cells in that pig will have those genetic modifications. So if you're looking for a liver, that liver has those modifications. If you're looking for a heart, that heart's got those modifications. And you can then use that heart or that liver, that organ, and transplant that into a human patient who needs that organ for survival. So it's still very much an experimental procedure. They've done it in non-human primates, which are the closest approximation in the animal kingdom to humans that we can get our hands on. And some of these organs can live for nearly 3 years. The trouble is that you can overcome the hyperacute immune response, but there's still the adaptive immune response. So the recipient of the organ still has to be on a powerful cocktail of immunosuppressant drugs. And even then, the organ is still under attack. And furthermore, pigs have viruses. We have viruses. You could be introducing a pig virus into the human when you introduce that organ. So I decided to put this into the slides because it was in the news in March when I was preparing the presentation about how a person had received the first ever liver transplant. And it was from a genetically engineered mini pig. 6 genes were engineered, and that was to remove those antigens I was talking about before. And they screened it for pig viruses, and they inserted it into a 50-year-old man who was clinically dead. I guess, they had the family's permission, the person was clinically dead, so there's perhaps less ethical issues than inserting into someone who's alive. And they just wanted to see how well that liver was going to function. And so they did it for, I can't remember how long it was, for 30 days or something like that. And they checked that the liver was functioning normally and the cells were healthy and everything was okay. And then they disconnected the liver and the person was dead anyway. So I guess that boggles the mind as to how to define death, which is another question. But anyway, so this has previously been done with pig hearts and kidneys. So David Bennett is an American who received a pig heart with 10 genetic modifications, and he survived for 60 days. So he was refused to be a recipient of a human heart, apparently because he had a record of disobeying doctor's orders, and somehow or other, I guess because hearts are in short supply, that meant that he was down the bottom of the list for recipients. So he was doomed to die as a consequence of ignoring his doctor, so let that be a lesson to all of you. I ignore my doctor from time to time, I have to confess. And he said when he was offered this experimental trial that he wanted to live. So he signed up, he wanted to live. And so he lived for long enough to be able to watch the Super Bowl, and I think there was a YouTube video of him watching the Super Bowl at the Maryland hospital where he received the heart. But he only lived for 60 days because unfortunately the heart he received had a pig virus in it. And because he was highly immunosuppressed and there was this virus, they couldn't balance those two. If you don't have an immune system, you can't destroy the virus, but you can't have an immune system because it's going to destroy your heart, so then you're stuffed. So he only survived for 60 days. But there is another option, and it's very much still only being researched. It's not happening now, and I don't know whether it will ever happen, but it could. And this option is to create human monkey chimeras or pig human chimeras. And what's a chimera? So a chimera is an organism which has 2 independent genomes. So on very rare occasions, human chimeras can exist if 2 embryos merge early in development. And then you actually grow into a normal adult person, but you have the genome of 2 of you, you and your twin, or your twins and you. So some of your cells have that person's genome and some of your cells have that other person's genome. And so you have 2 genomes within an organism. That's a chimera. And that's fairly routine in some places that work with genetically modified animals. You can take an embryo and introduce stem cells into that embryo, and then those stem cells will contribute to the development of the embryo, and the organism which results from that has some of its body cells that are derived from the original host embryo, and some of the body cells are derived from the genome, have the genome of the cells that you introduced into the embryo. So you end up with an animal with 2 different genomes. So if you have a pig embryo and you introduce human stem cells into that embryo, then you can develop an animal which has some of its cells derived from the human stem cells that you put in. So it's a human pig chimera. So at the moment, the chimerism is the human cells kind of get in the way. There's a bit of a species barrier there. Chimeras can be generated. I think the longest that they've lived is just an embryo that goes to 4 weeks of age. So they haven't actually made the animal go to term. So 4 weeks of development, I should say. And because the human cells are not so great in the pig environment, they don't contribute very much to the body of that animal. But in theory, in the future, if we learn a bit more about why human cells don't do well in a pig environment, we could perhaps genetically modify them or provide them with the right environment so that they can grow. And I've already spoken for my allotted time, and I'm only 2/3 of the way through. There's a potential for, you could make a genetic modification in those pig cells. Say you're looking for a heart, and it's a gene which is essential for embryonic development of the heart. And if that gene is deleted, the pig embryo will not develop a heart. But you provide human stem cells that don't have that gene deleted, and then you could have a pig which develops a human heart within that pig animal. And even better, if those stem cells are derived from your body, that would be your heart with your genome growing inside that pig, and that would be perfect for you to receive as a recipient. You have no immune response at all against that heart. But it potentially raises issues, and I don't want to discuss it too much, but one thing is that there's no clear dividing line on the question of overly human chimeric brains, because we lack an understanding of what point humanization of an animal brain could lead to more human-like thought or consciousness. We don't even know what happens in a normal developing human brain, let alone in like a chimeric situation. So people envisage this Frankenstein scenario where a pig develops and some of the brain comes from the human cells and some comes from the pig cells, and you have this human-pig chimeric brain. What does that look like for that organism? So nobody knows. But it's not been done yet, and it's in the future maybe. So genetic engineering holds potential for treatment of diseases and for xenotransplantation, but people say all sorts of things about human genome engineering, like before I mentioned designer babies. I want my baby to be intelligent and witty and have blue eyes and blonde hair and be socially outgoing and stuff. But there are lots of limitations to genome editing, which may mean that never actually comes about unless we're able to overcome those barriers. So one of the things is delivering the CRISPR-Cas9 genome editing components to the organism. So at the moment we could use viral techniques, electroporation, microinjection, nanoparticles, but they're all kind of limited in an actual organism. If you look at the example of the sickle cell disease, it was done in a cell culture, hematopoietic stem cells. If you look at the example of He Jiankui and the babies that are genetically engineered, it was done in cells. It was done in an embryo of 8 cells. And in the pig xenotransplantation, it started with a pig skin cell that was genetically modified. It's easy enough to introduce the CRISPR genetic components into a cell. It's very difficult to introduce it into my biceps. I've got a muscle wasting disease in my biceps. How the heck am I going to get all of the cells in all of my muscles throughout my whole body to be genetically modified so that I have normal muscles? It's very difficult. We don't know how to do that. Maybe we could have a virus which specifically invades muscle cells and introduces the CRISPR-Cas9 genome editing into muscle cells. It's not going to be 100% efficient. And we don't have such a virus as far as I'm aware at this point in time. So that's one limitation. The other limitation is, as I was describing before, we don't fully understand how the genome functions. There are a number of monogenetic diseases, and by monogenetic, one gene causes that disease. If you have an extension of a repeat sequence within the Huntington gene, you will have Huntington's disease. If you have a point mutation in the gene which is responsible for Duchenne muscular dystrophy, you will have Duchenne muscular dystrophy. But what is it that makes you intelligent? Is it that gene, that gene? Is it 1,000 genes? Is it the interaction of 10,000 genes? Is it 30% to do with your genes and 70% to do with your environment? You went to a good school, you had good parents, you ate good food. What is it that makes you intelligent? What is it that makes you witty and outgoing? I can't tell you how to genetically modify an embryo to make that embryo intelligent. Something like sexual orientation, maybe certain genes predispose a person to being homosexual, but which we've kind of got an idea as to which ones may help to predispose it, but you could perhaps edit those, but that's not going to guarantee what that person's sexual orientation is. Perhaps a bad example, but the point is, for some things, yes, it's obvious, that gene does that. But for the majority of things, it's not obvious at all. So I can't give you a designer baby simply because I don't know how, right? How do you make a person more compassionate, more intelligent, or things like that? So genetic systems are complex, interacting, and adaptive. So we don't know what effect genetic modification at one point will have on the rest of the system. and genes typically have more than one function and interact with multiple other genes. Another limitation of genome editing, as I've already discussed, I think, with the girls that had their genome edited, is that there are on-target and off-target effects. CRISPR-Cas9 cuts on target, that's great, but how will that cut be repaired? You could introduce any number of mutations which could in fact impact that gene and surrounding genes in different ways. Or it could cut in rare instances, but still it's going to be biologically relevant potentially in an actual human being. At another location in the genome, you have no idea where. What impact will that have on people? So is it safe, right? So our knowledge of genome function is limited, and editing the genome could have unpredicted consequences. So I think that sort of at this point in time limits the potential. People, talk about various things, but will that really ever happen? Who knows? So what did people think about genome engineering? So the University of Melbourne conducted a survey. They advertised it on social media, and they put up flyers around campus. So their methodology for collecting participants in the survey perhaps could, leaves a little bit to be desired. But still, it's an Australian survey, and the only one I could find in Australia. So it was a web-based questionnaire. There were 1,500 respondents, and they came from all different countries. I guess, University of Melbourne has got a few contacts, predominantly with a tertiary degree, which I suppose is not surprising, given that it was advertised throughout the university. And they asked respondents what extent they supported different applications of germline genome editing. And generally, respondents were supportive, particularly if the application was medicinal. But opposition was higher if the application was enhancement. I just want to look better, or I want to be smarter, or things like that. And also, enhancement in healthy individuals, so it wasn't treating a disease in any way. And I don't know if I've said it here, but they also found that there was a lot more support if it was just somatic editing, so not passed to the germline or to your descendants, but if it was just in that individual who is suffering from the disease, a lot more support for it in that case. So the greatest opposition they found was amongst religious respondents and also those with experience in genetics, which was interesting because maybe some people with experience in genetics might understand some of those potential limitations or drawbacks a little bit better than the general public might. When the general public hears, oh, you'll be healed of this disease, oh, but the risks are, right? So what does the scientific community say? Well, the scientific community says heritable human genome editing should not be used unless, at minimum, it meets reasonable standards for safety and efficacy and is legally sanctioned, and has been developed and tested under a system of rigorous oversight that is subject to responsible governance. And remarkable progress has been made in somatic human genome editing, demonstrated it can cure once incurable diseases. To realise its full therapeutic potential, research is needed to expand the range of diseases it can treat and to better understand the risks and unintended effects. The extremely high costs of the current somatic gene editing therapies are unsustainable, so a global commitment is needed for affordable, equitable access to these treatments. So, that sounds reasonable and responsible to me. But the thing about it is that it says it has to be safe, it has to work, and it has to be permissible, right? It has to be legal. Which I guess is reasonable for a bunch of scientists just to focus on those three things. But is it right? Is it ethical? What are the social consequences, the political consequences? What about people's religious or theological objections? Not covered or considered by these statements, so perhaps we need a broader debate. But how is that debate had? And if, let's say the scientific community decides that, yeah, it's safe, yeah, it's not illegal, yeah, it works, we'll just do it. Well, who actually regulates the scientific community? In China, this dude just went and did it because he wanted to do it, and then as Christians, perhaps we have additional ethical concerns or theological concerns. Are we playing God by modifying the human genome? Here's a quote from Paul Ramsey. Man ought not to play God before they learn to be men, and after they have learned to be men, they will know not to play God. So, although we are creative by nature, we're not ultimately the creator. So does that mean that there are boundaries that we should not cross? But what are those boundaries, if there are boundaries, and how do we know what they are? And then the question of violating the image of God in humanity. So Francis Collins, who led the public genome sequencing effort in the United States and is the founder of the Biologos organization, says, but from a theological perspective, if we are fearfully and wonderfully made, do we want to be fearfully and wonderfully remade in some other kind of image based on what we think we should become? So I thought that was an interesting statement. So I wanted to look a little bit more at the image of God. So we're probably all aware of Genesis 1, where God said, let us make mankind in our image and in our likeness, so that they may rule over the fish in the sea and the birds in the sky, over the livestock and over the wild animals, and over all the creatures that move along the ground. So we're familiar with that creation story in Genesis, and then it's also mentioned in the other creation story in Genesis 5. When God created mankind, he made them in the likeness of God. He created them male and female and blessed them, and he named them mankind when they were created. When Adam had lived for 130 years, he had a son in his own likeness and in his own image, and he named him Seth. And then we see the image of God also in the New Testament. In Hebrews, the author of Hebrews writes, God, having in the past spoken to the fathers through the prophets, and many times and in various ways, has at the end of these days spoken to us through his Son, whom he has appointed the heir of all things, through whom he has made the worlds. His Son is the radiance of his glory, the very image of his substance. Corinthians 1, Paul writes, the Son is the image of the invisible God, the firstborn of all creation. For in him all things were created, things in heaven and earth, visible and invisible, whether thrones or powers or rulers or authorities. All things have been created through him and for him. He is before all things, and in him all things hold together. He is the head of his body, the church. And then in Romans, Paul also states that we are being, as Christians, being conformed to the image of Christ. Because those whom he has foreknown, he has also predestined to be conformed to the image of his Son, that he might be the firstborn among many brothers. So what does it mean, this image of God idea in Christian theology? Well, one could say that it asserts the dignity of humanity, and it's a special honour bestowed to humanity which sets us apart from the rest of creation. But what is it exactly referring to when we say we're made in God's image? How is God's image manifest in us? How do we know, or how can we see that we're made in God's image? And so it's been debated a lot throughout Christian history, and so there's a lot of ideas as to what this means. And so I'm not a theologian, but I kind of got the impression that there are different ways of grouping these ideas. One is the relational view, that the image of God in us mirrors the relational nature of the Trinity, God the Son, Father, and Holy Spirit, in constant communion. Humanity granted the free will necessary to have relationships. The ability to maintain and establish relationships with God and with others is how we as humans display the image of God. Then there's the substantive view. That is that humanity shares attributes with God. And maybe that's one you hear a lot, I don't know. But it's things like, humans are rational and we have intellect. And so God is rational and has an intellect. Humans have morality, God has morality. Humans are creative, God is creative. And this idea of spiritual growth, as we develop God's moral character, we better model his image. So it's sort of like, what are the unique characteristics of humanity that set us apart from the rest of creation? Our ability to love others, our ability to be rational, our creativity, our morality. That's the relational and substantive view. There's also the functional view, that what it means to be made in God's image is to be God's representatives within creation or within the creative order. And I think it's important I personally think it's important to remember that Genesis is the product of an ancient Near Eastern culture. And so when interpreting Scripture, I think it's important to keep the historical context of the passage in mind and the cultural context of the passage. So when one thinks that in the ancient Near Eastern culture, you have empires, the Babylonian Empire, Persian Empire, whatsoever, where there's a king who has to rule over the empire, but it's difficult to travel throughout the empire. You can't just hop in a plane or in a car and be there and assert your authority. You can't send emails. So what you do is you set up your image statues of yourself throughout your empire that says, this is my turf, I'm the boss here, right? So kings would set up images of themselves where they wish to establish and maintain their authority and rule by divine mandate, because kings in empires often said, I am a god, you will do what I tell you. Or at the very least, I am the son of a god, or like a god, you will do what I tell you. And perhaps the Israelites were different in that regard, where they always acknowledged God as the ultimate authority in their society. Well, that was the ideal. They didn't do it very well very often, but that was the idea of being a theocracy. So kings ruled by a divine mandate and had dominion over creation, over their kingdom. And likewise, we see in Genesis 1 there that the image and likeness of God is straight away associated with you will have dominion over and rule over the beasts of the earth and the field and blah, 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 right? So there's this association of authority. And then we see in the New Testament where it talks about Jesus having the image of God, it then goes, for he is creator of everything, the boss of everything, right? He is the head of the church, his body. So again, the image of God is associated with Christ's authority in that passage. So one could say that Jesus is the ultimate image bearer of God, because we display God's image or God's authority imperfectly because of our sinful nature, whereas Jesus modelled the image of God in a perfect way. And so an argument could perhaps be made that while the Old Testament might focus on the image of God being related to God's authority, and the New Testament has that element too, the New Testament also introduces another element where the image of God for us as Christians is to become more and more Christ-like in Christ's character and in God's action or activity in the world in terms of perhaps, restorative justice and things like that. So N.T. Wright therefore says that when humanity brings about God's restorative justice, we reflect God into the world and become God's image. And Peter N. says, Christians are now full representatives of God in his creation, but not in the ancient Near Eastern sense or the Old Testament sense as being a ruler. And not that he's dismissing that, but being a ruler is not the New Testament emphasis. The emphasis has now moved to other things. Christians represent God to all creation through modelling humility, love, and holiness. So the more we take on Jesus' character and approximate his image, the better we are reflecting the image of God in creation. So that's the functional view of the image of God. And then there's the physical view. And I don't think we want to take a Gnostic perspective where we say, all flesh is bad, all flesh is sinful, because the body is God's temple. The body is God's handiwork. We're fearfully and wonderfully made. Jesus died in the body and was resurrected in the body. Jesus has redeemed our physical bodies, and Jesus had a physical form. So it's not that our bodies should be treated as bad or ignored. They're good, but I'm not sure that captures what is meant by the image of God. You see, because I have an appendix, does that mean God has an appendix? It becomes, I think, problematic in that regard. And then remember that we are a product of evolution, like every other living organism on this planet. When did we approximate the image of God in our evolution? Was it a sudden thing or a gradual thing? Are chimpanzees close enough to have the image of God or not? So for me, it's a little bit problematic to think of our physical identity as reflecting the image of God, whereas I would think personally that's something like the other views which are espoused perhaps are less problematic for me. And I sometimes think that, we're bipedal apes that became aware of God's presence and are able to have a relationship with God. But what if God revealed himself to some sort of cephalopod? This is a really crazy question. And then would the cephalopod who has a relationship with God say, I'm made in God's image, what wonderful tentacles I have. God has fantastic tentacles to shepherd his children. So, for me, it's awkward. But that's not to say that people being physically made people's physical nature doesn't have moral implications. Because it does say in the Bible, you should not murder because humanity carries the image of God. You should not curse humans who have been made in God's likeness, right? So then, what are do the moral implications extend further than slander and murder? Do they extend to gene therapy? You should not cure your incurable sickle cell disease because you are made in the image of God. Well, I don't know. I go to the doctor for all sorts of things. I'll happily take a medicine to treat a disease, right? If I have somatic gene editing, it's only going to affect me. What about cosmetics and surgery? We alter our appearance. You know, if my ankle is broken, I get it altered to fix it. If my genome is broken, should I get it altered to fix it, right? So maybe I sometimes think that the Christian approach to genome editing is always, oh, this is disturbing, this is bad, we shouldn't be doing this. What about this concern? What about that concern? What about this ethical implication? Right? And I think it's fair and right that we have a discussion about these concerns and these ethical implications, but sometimes it's the only thing we ever talk about. Whereas what about the person who is suffering from an incurable childhood illness, and they've suffered all their lives from that, and now they can be healed? What about the woman who was bleeding all her life and she touched Jesus' cloak and she was healed? Right? So I think maybe there's another aspect to this debate which is not often heard in Christian circles. What about the imperative to do good? So in Matthew 25, Jesus says, when the Son of Man comes in his glory and all the angels with him, he will sit on his glorious throne, and the nations will be gathered before him, and he will separate the people one from another, as the shepherd separates the sheep from the goats. He will put the sheep on his right and the goats on his left. And the king will say to those on his right, come, you who are blessed by my Father, take your inheritance, the kingdom prepared for you since the creation of the world. For I was hungry, and you gave me something to eat. I was thirsty, and you gave me something to drink. I was a stranger, and you invited me in. I needed clothes, and you clothed me. I was sick, and you looked after me. I was in prison, and you visited me. On what basis in this passage are the sheep separated from the goats, followers of Christ from those who do not follow Christ's example? I hate to say it, but it's a works-based passage. It's not because they said the sinner's prayer, and they had the right theology, and they ticked all the boxes in the quiz and got all the answers right. It was because they fed the hungry, they visited the sick. It was how they treated those less fortunate to them. And likewise, those who did not do so were the goats on the left. So Jesus is separating people on the basis of, right? So maybe there is, if in following Christ's example, where he went around healing the sick, and he speaks this parable, for us as Christians to do likewise. So in addition to voicing concerns and exercising cautions, should Christians not be equally excited about the opportunity for the healing of disease that genome editing offers? Christianities will often refer to modern medical intervention, doctors, nurses, etc., as a gift from God. There's a person in my church at the moment who has Guillain-Barré syndrome. His immune system is attacking his nervous system. He's in a coma. He's hooked up to machines so he can breathe, so his heart can beat, so his bodily functions keep going, because his brain can't regulate anything his body's doing because his nerves aren't talking to anything. The only reason he's alive at the moment, and who knows how long he may stay that way, but by God's grace he will not stay that way forever, is because of modern medical intervention. Right? So can we not consider modern and the amount of resources poured into keeping this poor man alive? It's incredible that we are blessed in a country like Australia to be able to devote these resources to treatment of these diseases. So should we not, as Christians, view the potential to do good using this technology as a potential gift from God? Shouldn't Christians be in the business of alleviating human suffering? So, hopefully all of that's given you something to think about, and hopefully you have enjoyed it as much as I enjoyed putting it together. But I want to leave you with a few guidelines. And I think technology places power in the hands of fallen humanity, who then use this technology for good and for bad. And we see that in so many different aspects of technology, where the more technology we gain, the more powerful we become, and the more severe the consequences are of us using that technology. And yet we're a fallen species and we don't have necessarily the wisdom or the maturity to be able to always apply that technology in the right way, especially when we are driven by greed and the profit motive. If CRISPR-Cas9 genome engineering is being driven by a desire to heal people, maybe that's well and good, but it's being driven by a desire to simply make profit. Perhaps that won't always lead to a good outcome. So 3 questions from Trevor, can't read the surname, sorry, that maybe help us to navigate this, or maybe just muddy the waters even further. That is, in any given treatment, and I think they sort of need to be considered on their individual merits, does it increase the ability to view life as a gift to be received with thanks or a commodity to be grasped as a right? Does it acknowledge human nature as something to be restored to wholeness or something which is to be transcended by enhancement? Does it reflect wise stewardship and care for the world as creatures made in the image of God, or does it seek to enable us to become as God in recreating ourselves into something in our own image? So I'll leave you with those thoughts, and thanks for putting up with me talking for such a long time.
Thank you, Jonathan. That was fantastic, I think.
Thank you.
I started out thinking I'm probably against any messing around with the human genome, and now I'm totally not so sure.
Neither am I.
A lot of food for thought there. Thank you very much. Well, I think we've got plenty of time for questions. There's a microphone here. I'm going to pass it around to anyone who would like to ask a question. We're kind of recording this, so it's as much for the recording as for everyone else's benefit. Who has a question?
Really formed my question yet, but I consider the purpose of the Holy Spirit by which Jesus healed.
Yeah.
And if we are made in the image and likeness of God and to walk in the Spirit and to do as He did,
Mm-hmm.
is this not the way that we should first consider healing? Is this not the way that, not to be transformed by the world, but by the renewing of our mind to the truth?
Yeah.
And walking in that Spirit as Jesus did. Like you gave the example of the woman with the blood issue. He felt the power go out of him. Yeah. Into her, and that's what healed her. Her faith healed her.
Yeah.
She received because she believed. There seems to be in that a definite clarity of instruction, whereas all that you've given tonight, which was amazing, and thank you so much. What would be the purpose of having a Holy Spirit if we're using the means of the world to heal?
Yeah, I mean, that is a difficult question for me because, maybe I haven't been gifted with sufficient faith, but this person that I spoke about who has Guillain-Barré syndrome, if he collapsed in the shower, and if he hadn't gone to hospital, he would be dead now. And the cynical part of me says, well, that they could have prayed for him, but I've seen too many examples where a Christian family has a child with cancer and the church prays for that family, but they still go and get medical treatment and the child still dies. So I don't know personally if healing is something which was limited to Christ's ministry, or physical healing is still a thing today. And I think God has given us, we're fortunate enough to live in this society which has this technology, that we should avail of it. Which is not to say that it's perfect in any way. There's a lot of things that we don't understand. There's a lot of places where medical technology doesn't help people and can make people worse. It's difficult.
I didn't expect you to have the answer, but I really appreciate your part in it and your
I might just comment on that just briefly, and then I've got a sort of a question related to the talk. It's a little bit different. I've also had a look into justifying sort of physical healing and spiritual healing, and it seems to be that Jesus used both as well. So there's him being touched, but there's also him spitting into the dirt and mixing it up and putting it on people, and it just seems really sort of, and when he did his healing, it wasn't sort of like, he didn't do it like a magician. It wasn't like a show. It wasn't sort of like, look how much power I have over everything. It was often using the physical things around him. Like it was using he didn't just magically make the food come out of nowhere. It was the food was already there, and then it was duplicated, So it's often it doesn't try to usurp the physical world. It tries to sort of enhance it and extend it. And I think Christians throughout history have done that with their approach to health and healing. They've prayed for the sick, but they've also understood the natural world, and like Jesus kind of had this sort of holistic approach to healing. And I had just talked to a missionary who they were a medical doctor, and they would use drugs to heal, but then they would pray for them afterwards. And so there was this sort of connection between the spiritual and also the physical healing. So just a short comment about that.
Yeah, thanks, Jake. That's good. I think, maybe being as a scientist, it's too easy to be a reductionist and go, there's nothing but the physical world. But I think it's more fulfilling to think of humans holistically as having, a spiritual dimension, perhaps, and that a sick person can be treated with medicine physically, but there's something about that person. Maybe it doesn't supernaturally heal them or something, but they also need to be prayed for, just because they're human. I don't know. Yeah.
No, that's yeah. That's a whole other talk. My question's about the image of God. So I really enjoyed the sort of description of that. And I was sort of reminded of that quote from the beginning on human rights from UNESCO.
Yeah.
And I just was thinking that UNESCO quote almost puts the human genome in the position of the image of God. In the sense that it's like, this is something that unites us all, and it's the thing that makes us.
And it's our common heritage.
It's our common. It's like a uniting thing. Like human rights is supposed to be that sort of uniting principle, but often without having the spiritual aspect or the purpose behind the object of the rights, you kind of it's kind of just sitting there in the air, and you're not quite sure what to do with it until you know, like, what's the spiritual purpose of it. And I was just sort of wondering whether you'd kind of had a thought about how the image of God, that theology, links to how, like, the people, the scientists and the ethicists sort of talk about it sort of as this sort of dissociated from that purpose and spirituality.
I'm not sure I quite follow. Sorry. Yeah. It's an interesting thing because, maybe coming back to reductionism again, what is it that makes you human? From a purely biological point of view, your genome would have to be a significant contributing factor. It's not the only contributing factor, but it would have to be a significant contributing factor. But the same thing, it seems sort of hollow to say, well, all I am is the sum of my genes. It's like saying, when I love my spouse, it's because I have these chemical reactions in my brain. There's that one level of explanation, that mechanistic level of explanation, where they're always using that example of the kettle's boiling. Why is the kettle boiling? Oh, because the energy, the enthalpy and the entropy in the molecules is causing the water to change state and it's boiling. But it's also because Auntie Ethne wants to have a cup of tea. So, I find it difficult to scientists can get, I think, trapped into this purely materialistic view of things, and somehow that that denies something of the full richness of human experience.
Thanks. So I guess there's already been a lot of talk about the ethics of gene editing and things in humans. I wonder if there's been any sort of thought among Christians about these sort of procedures and techniques in the rest of creation, as in the animals, especially maybe the higher order animals, where we're using them for experiments for these sort of things like pigs and
Yeah.
apes or whatever. So has there been any Christian thinking about this as well?
Well, I guess it's not something I looked into in preparing this talk, because I specifically was looking at human applications. But I would imagine that yes, there has been, because there was certainly some Christian discussion about genetic modification, and we know that it's a topical thing, especially when you look at the animal rights movement in a lot of countries. Some of the more militant or active vegans and things like that. It certainly is an issue, the treatment of animals in all fields of science, not just genetic modification. Which is why, in Australia, we try to have a balance between the benefits of using animals and the perceived drawbacks of animal suffering or violation of animals. Because, the reality is that a lot of the medical treatments which we perhaps take for granted took decades of research and investment, but they also all had a translation from the lab to the clinic, often through an animal model, to ensure it was safe and effective. So, we can't have the benefits of these advances without also potentially these ethical issues being involved. So I guess to answer your question, I didn't look into it, but I imagine that there is, talk about it. But Christian theology, I think, has always placed humans in a separate category to the rest of nature, in that because we are made in God's image and because we have received the breath of God and we were made on a separate day, and there's theological reasons for thinking that humans alone are God's children, whereas the animals don't partake in that. So therefore, it's not quite the same theologically or ethically from a Christian perspective.
Just thought to add something to a perspective to this question before about Jesus healing by the power of the Holy Spirit, and there is this, what we heard tonight, as if that has nothing to do with the Holy Spirit, and what I would, it's not an answer, it's just a thought and a perspective. There are a great deal of Christian scientists whom God has destined and purposed to become Christian scientists, come into medical field, work on genetics and DNA in the medicine, and from their birth there has been a Holy Spirit guiding them along this path. And as they stepped into academia and got their degrees and further into the industry, they have had an ongoing relationship and guidance, and by the power of the Holy Spirit, they have obtained degrees and insights and revelations and ideas and solutions. And maybe this idea, many of these ideas, they may have received from the Holy Spirit. So I think we cannot look at this is the miracle of healing of Jesus, this is the work of the Holy Spirit, but us here, healing it's the work of man. I would disagree with that, because what we are studying human beings, human bodies, the way how they are created. And we are working with the healing mechanisms that are already in place in our bodies. So when we are, the doctor this is a miracle. Even when our wounds heal, it's superficial wound, it can heal itself. It's still a miracle if you think about it. And so the doctors are just studying these existing healing mechanisms in place and trying to cooperate with some things that God has already put in place. And many of these ideas are Holy Spirit inspired. So I think we cannot afford to see all this as a work of man, because God is in that. It's just his different way of engaging with him, loving us and engaging with us in every moment of our lives, in everything that we are doing. Yeah.
Mine's just a little comment, but just was struck by your comment about hubris. And whenever I like to think that we as humans are pretty clever, I remind myself that a huge amount of our genome that makes us humans is actually from viruses, has that base and that commonality, and that we share a lot of our genome with a lot of the rest of the created world, and we're by far and away not the most complicated organism on the planet from that point of view, I think.
Yeah, that's true. Especially when you consider plants and how many genes they have. Yeah. So it seems like there's very little to do. There's not a whole lot of correlation between the complexity, however you define that, of an organism and the number of genes. Yeah, it's probably more a question of how you use it rather than what you've got. Yeah.
Can I butt in with one? I've been busting to ask this one, and then we'll get over to you. Hopefully a reasonably quick one. I'm just interested in how much Christian input gets into the sort of ethics discussions and ethics committees and whatever. And then the next question, kind of a separate question as well. From your last slides, I was thinking, I think you're distinguishing between restoring wholeness and enhancement of humans or the human genome.
Yeah.
Do you think the sort of Christian consensus will be to restore wholeness and stop there and not go with the enhancement?
Yeah, well, I mean, it seems that that's largely where it falls at the moment when you look at people's, what they're writing and that public opinion survey. It seems reasonably clear-cut that people are happy with the idea of correcting genetic disorders that cause disease, but are less comfortable with the idea of enhancement. However, you can actually kind of almost put forward a theological argument for pretty much anything. So I don't know whether it necessarily will stop there. You could say that, you could make an argument to say that, maybe we're doing God's work by restoring humanity to its ideal as it was before the fall, or something like that. I don't know, but yeah, I think broadly speaking, that seems to be where public opinion falls at the moment. Is we'll just stop with therapy for treating disease. But the thing is, I think society will quickly make the argument if it becomes feasible, because I don't think it's feasible at the moment. But if it becomes feasible for that enhancement as a reality, and if there's money to be made in it, a millionaire wants to change their eye color or something. The millionaire will simply say, well, why not? It doesn't affect you. It doesn't affect my children. Why can you tell me what not to do? And I'm willing to pay $1 billion to get it done. And you think you'll find society will very quickly allow that because the money is there. I don't know. I look at society and what drives us as humanity. Maybe this is a cynical and simplified view of humanity, but often it's the profit motive. If there's money to be made in an endeavour, humanity will undertake that endeavour.
Which I think sort of begs my first question. Do we desperately need more Christians having input into this ethical debate, do you think?
Well, I mean, I think the correct answer to say is we need all people in society to have input in this debate. But obviously, as Christians, we would definitely like to have Christians have an input into this debate. The question is, what is the avenue? Because if you get a bunch of scientists together to discuss this technology, they will invariably focus on the science because that's what their job is. I've been to scientific conference talking about this kind of technology, and they'll talk about the techniques they use and what they're doing or what their results are. No one will say, well, hang on, is this the right thing to be doing or not? Because that's not what scientists are there for. And I don't know where the bioethicists are coming from, but there's all sorts of different branches of ethics. And there's plenty of ways of viewing ethics that are not Christian ways of viewing ethics. And how does society decide what should be done and what shouldn't be done anyway? Like, when it comes to genetic testing on animals, it's only activism, really. I personally don't have much of a problem with it, but lots of people do have a problem with it. They seek to influence politicians through activism because it's not the sort of thing where it's like, oh, here's a referendum or a plebiscite or whatever it's called in Australia, where what's your opinion on this? You're not going to be asked, and a politician's not going to campaign on the basis of genome editing or something like that. They're going to be campaigning on the basis of goodness only knows what. The economy and jobs and healthcare, or maybe whatever fear-mongering or mudslinging they can come up with. But they won't be campaigning on that, maybe except for the Greens or something like that, or a niche party. So I don't know, what is the avenue by which Christians or other people, or other interest groups in society inform policy? It's only through our elected representatives and whatever committees come together to make these laws. I don't know. So, yeah, but the thing is, and I think that's a valid point. And of course, there's no reason why Christians can't and shouldn't be activists. But with many things in Christianity, what is the Christian position? Because their Christianity is a spectrum of beliefs.
Yeah, this wasn't part of my question originally, but there is already clinics available where you can screen your embryos and choose if your child has blue eyes.
Yeah.
And
is there? I know you can get screening of
US.
embryos for genetic diseases, but do they offer it for cosmetics?
I'm pretty sure in the US somewhere there is. I did my bachelor's degree in genetics, and we were talking about it.
Okay, yeah. It's certainly, it's probably possible. There's not too many genes involved in eye colour. So you could, the technology is there.
Yeah, and if you have enough money, you can. If you
Probably, yeah. If the country you live in doesn't...
Your baby to have blue eyes, you could have a baby with blue eyes.
Yeah,
Guaranteed.
and of course the thing which I was going to mention but I failed to mention in my presentation is that, with germline editing like that, you won't just edit a single embryo, you'll have any number of embryos to ensure that one of the embryos has the genetic modifications you want. So, if you're screening multiple embryos and 5 of them are brown eyes and 2 of them are blue eyes, well, what do you do with the 5? You throw them in the bin, right? So then that has additional ethical implications for those of us who believe that life begins at conception, or the unique human identity is forged at conception, because they would then consider those embryos to be human beings. Whereas, that's less of an ethical issue for those of us who don't consider that unique identity is conferred at conception. But, you see it again, another ethical issue to consider.
Yeah, definitely. But my original question was actually about what, I guess, the ethical and Christian perspective is on gene drives, where you would propagate a gene throughout an entire species.
Yeah.
Like with the malaria example, propagating malaria resistance in mosquitoes, or even wiping out all mosquitoes altogether by propagating all of their offspring to be females and not being able to reproduce anymore.
Yeah.
Is that not playing God?
You see, I didn't talk about playing God in this presentation specifically because to me it's such an amorphous concept, and it would be interesting to investigate it more to see if I can put any substance to it, because how do you define what playing God is? And don't we by our very nature play God all the time? So if we were to use a gene drive to intentionally drive a species to extinction, it would be by no means the first species that humanity has driven to extinction. The difference would be that we've always done it unintentionally before, except perhaps for Tasmanian Aboriginals and other things like that. But yeah, I mean, is it playing God? But then again, we've been given dominion over creation. Or does that mean responsible stewardship? Right? So again, it's a theological viewpoint. The thing is that you could by eradicating the mosquito that carries the Plasmodium malaria parasite, you'd eliminate malaria. And I don't know, it's the biggest killer of humanity outside of like heart disease and cancer, malaria. So then if you were a utilitarian in terms of ethics, maybe you would go that the good outweighs the bad. And of course we should remove the mosquito from the environment. There are other mosquitoes that can fill that ecological niche perhaps. It won't have a significant impact if we remove one of them. But yeah, I mean, who gives us the right to do these things? And yet, as humans, we modify our environment to the extent that we have driven endless numbers of species extinct and many others to the brink of extinction, and continue to do so, and will always continue to do so. So, does that make it right? Just because that's what we do, does that justify it?
And at what point, like you were saying, chimeras with like pig-human brains?
Yeah, that's a little bit science fiction, but they are working on it.
At what point is an animal too smart, I guess, as well, or like too close to us, too human, or like too close to us even, like a family dog or something like that. Or people say pigs are very smart as well, and that sort of thing too. Like, yeah, exactly.
Yeah, I mean, there's no question that animals are.
Yeah, exactly. How a pig could have human consciousness, but do they have human vocal cords that could say that they have?
There's no questions that pigs are that pigs and other animals are intelligent, but maybe one can argue that if intelligence is a spectrum, that humans occupy one end of that spectrum, but that maybe that's not where the image of God lies. Maybe it's our ability to relate to God that makes us special and separates us from the rest of the animals. Or maybe it's simply because God says you are in my image and not the pig. So I do think that Christian theology does draw a line in the sand between humans and other animals, which is not to say that we're not part of evolution like everything else. We are part of creation like everything else. But theology draws this artificial line because God says so, almost. Yeah, I don't know. It's one of these questions that has no answer. I mean, because where do you draw the line? You do then end up becoming a vegetarian or a vegan or whatever. And if you want to take that position, go ahead. But I personally don't. So yeah. There's no answer. Sorry.
Okay. This is a lot lighter and easier, the question. You talked about I suppose the danger in the path. What happens when we do all those things, like the crazy guy in China that did that, and the unintended consequence and things like that? The question really is, how do you see that progress in, say, 20 years, 50 years, and how would it develop, and what kind of potential problems that we might get? Well, but at the back of that, I already had something which might kind of give us a glimpse. After this First World War and Second World War, there were lots of people, soldiers, that got their face half blown off. And a lot of plastic surgery was developed by doctors trying to fix people to restore part of their appearance and function of the body. Now, they did wonderful things, but then the technology progressed to today where we use it for other purposes, and we suffer the consequence of it too, because we can see disfigurement and all sorts of things happening. And that hasn't kind of fully played out, but at least that has 70 years of a time advantage, so we can see what goes down the track. Now, if you drew the parallel with gene technology, would you think it would be kind of self-limiting? In other words, it'll come to a point, you get bad consequence, and then people will kind of like, okay, no, we can't do that.
Yeah, I mean, it's always hard to look in a crystal ball and say what's going to happen in the future. But I guess we can learn from past experience. And I think you're right. As I said before, we have technology, but then we don't necessarily have the wisdom or the maturity to always apply it correctly. And we're always living with the consequences of that. And we see that in so many ways. Technology is enabling us to communicate with each other better than ever before, but then we also have young people who are addicted to technology and don't leave their bedrooms up gaming all night. Maybe that's a stupid example, but there seems to be always it's a double-edged sword. There's a positive, sometimes the technology is implemented with the best of intentions, but it can be applied in a different way as well. And I think sort of there's something about humanity that if it can be, or if it's beneficial to somebody to do so, they will do so. So, I mean, this CRISPR-Cas9 technology is constantly undergoing updates. The one I described in my talk is the classical system. There are many more applications and many different variants of that system now, which can be used to make all sorts of different genetic modifications. And the improvements are being made so that it's more and more safe and more and more effective. So I do think that, I mean, our understanding of the genome will probably improve with time, so that we'll have better understanding of the changes, what impact the changes we're making will have. Our ability to deliver the genetic modifying tools into bodies and cells may improve with time as well. So, I mean, in some ways, it could be a good thing if we reduce the incidence of some of these genetically inherited disorders. You could potentially envisage a world where people aren't born with that anymore, but can we? That's a very difficult thing to achieve, because that would mean that every single person who's born would have to have come from an embryo which has been screened for all possible genetic disorders. And that would mean that natural normal conception is no longer the done thing. I don't see that happening all of a sudden. And even if we did screen every single embryo for every single genetic disorder to make sure that people were only born without genetic disorders, there's still spontaneous mutations would arise from time to time. So it's kind of like this utopian idea that maybe could never actually be realized. But the possibility of introducing genetic mutations into the genome, which could have unintended harmful consequences, is very real. But then again, spontaneous mutations and natural selection and all these other processes are going on, introducing random variation into the genome all the time anyway. So it's not, in a way, it's not like we're doing anything new. It's just that instead of allowing nature to take its course, we're intentionally doing it to ourselves. So, I don't know if that answers your question, because like many of the questions, they're very difficult to answer.